So now what I have is, again, could you please close the door? Oh, I could even just give this tape to our administrators. Wonderful. Okay, so here I have our two centrioles. Remember, they're like tow trucks. But can you guys see right here that those centrioles, everybody look, that they cannot get to the chromosomes?
Yeah.
So what's in the way? What's this? This is made of phospholipid, another thing. What's this thing in the middle? The nucleus. So what envelopes the nucleus? Nuclear envelope or membrane. Well, this is in the way, so I don't have white out, but what you're going to see is this nuclear envelope begins to disappear, and the nucleolus begins to disappear. So here's the big three things. We have nuclear membrane breakdown, spindle fibers appear, and chromosomes condense. So now we're ready to move on. Does everybody go with that one? Yes? Lovely. Now from prophase, could somebody please answer my phone? Now from prophase, we are going to move on to metaphase. So, in metaphase, what we're going to do is let's start back up with the cell that we had. In this cell right here, okay, this is interruption number 2. What do they need?
He's going to keep Carter Skinner to finish inside Mr. Passman, if you don't mind.
Not at all. Okay. So, what we're going to do right here now is, the point is we need to make this into 2 different cells. Can everybody follow me? So if we're going to make this into 2 different cells, we need a copy of this chromatid in each new cell. So what do you think the spindle fibers are going to have to do to this? Pull them apart. Well, we already got the nucleus away. Now how should we pull them apart? Should I pull 2 copies of this on one side and then pull 2 copies of my mom's chromosome on the other side? Or would it make more sense to split everything in half? Okay, so this is what's going to happen is that all these chromosomes need to line up along the center. So what you're going to see here is, now notice what we don't have. Is there any nuclear envelope in there?
No.
But as these spindle fibers attach in the centrioles, you are going to notice, I'm going to attach that there. That was a very ugly one. That looks like a broken windshield. And I'll do a little one from Dad. And I'll do a big one from Mom. Now notice they could line up in any order. And a little one there. Okay. Now this is the easiest thing to determine. You can see, especially if I drew that one right, that all of the chromosomes have lined up along the center of the cell. Is that easy?
Yeah.
What do we think the next step would be? To separate them. Okay. So now we're going to move from prophase into anaphase. So in anaphase, what you're going to see is all of these chromosomes that have been lined up in the center are going to pull apart and go to the sides. The telltale sign is, notice they always point in the direction that they're moving. So you're going to see all of the chromosomes, the sister chromatid, are going to be separated. So again, here is our cell. You're going to see the large one from Dad going that way, the small one from Dad, the large one from Mom, and the small one from Mom. The large one from Dad, the small one from Dad, the large one from Mom, and the small one from Mom. Now notice how all of these, here's the one thing that is odd, is that it actually appears that these microtubules or spindle fibers are pulling them. It's actually not. On this, if you were to look really, really close, you're going to see that we have the centromere, which is that little spot in the middle. And on top of it, sitting right next to it, is a kinetic core. I believe that's how it's spelled. And the kinetic core is like 2 little feet. And honestly, it looks like 2 little feet. Like, you know, can a protein look like anything it wants to look like? It's made of protein. You feed it ATP and it goes blah, 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 and it runs along. So these things run along till they get to their individual size. That is going to be anaphase. So what do you think the next step would be? What do we need to do next?
Telophase.
Okay, now we're going to have telophase. Telophase, in an animal cell, these need to split. So what you're going to see is furrowing. And what actually is, just think if you had a string around a balloon here, and you started squeezing that string, what would the balloon do?
Pop.
Well, what would it do first? It would pinch. Well, that's exactly what happens. Is you have like microtubules, microfilaments along the center that start to pinch. And remember how we showed in vacuoles what happens when plasma membrane touches plasma membrane? They kind of fuse together. So as soon as you see this beginning to pinch, then you know you are in telophase. And that will continue to happen to the very end until you get to cytokinesis. When you have cytokinesis, you are going to have two cells chromosomes and then are going to unravel, and you're going to form your new nuclei around it, and then we're going to be right back where we started from, and you're going to have two cells that are exactly the same as your initial cell. That is the most common type of cell. That is mitosis, and that is for growth. Now, on your flashcards, each of these things that I covered, you need to make a flashcard about. Is everybody good? Questions, comments, concerns? Okay, so I will end this here.